Good afternoon, CGIL. This week, we are happy to have Dr. Pablo Fonseca, a postdoc fellow at the University of Leon, presenting for us. Dr. Fonseca obtained his Bachelor of Science in Biology at the Federal University of Minas Gerais in Brazil. Subsequently, he completed his PhD and MSc degree in genetics with an emphasis in genomics and bioinformatics at the same university. From 2017 to 2021, he joined Dr. Canovas here at CGIL, first as a visiting student in 2017 and later as a postdoc in 2018. Since 2022, he has joined the Mega UL group at the University of Leon as a postdoc through a grant from the European Union for the attraction of talents. Today, Dr. Fonseca will be presenting his research entitled Investigating the Genetic Basis of Feed Efficiency, Sustainability, and Resilience on Spanish Dairy Sheep through Omics Integration. So I'll turn it over to you now, Dr. Fonseca. Thank you.
Thank you very much, Gabriela. And first of all, I would like to say that I'm very happy to have the opportunity to present again a CGIL seminar. I remember the first time that I did it, and I'm still very happy to have this opportunity. So I hope that you like today the presentation. I'm going to skip this slide because Gabriela already did very well the introduction. And just to let you know a little bit, so Gabriela mentioned I'm from Brazil, from Belo Horizonte. It's a very nice city. I like a lot, a very big city. And I also had the opportunity to live for a while in Guelph, a place that I also loved. And currently I'm living in Leon, and another place that I'm enjoying a lot. So I'm very happy with the places that I live in this world. So I have nothing to complain. As a summary, today I will try to talk a little bit about, as you could see in the title of my presentation, a little bit about production, resilience, and also sustainability, but with a focus on some specific traits or some specific approach, such as trying to understand the genetic basis of feed efficiency, the relationship between production and health. I'll talk a little bit about pleiotropy, integration of selection sweeps, and if I have time, I will show some updates on the R package Gallo that I know that some people on CGIL are using very frequently, so I hope that I can talk a little bit about these updates. If not, you can also reach me always to talk about Gallo. I'm very happy to show the new codes or the new functions and help in any process that you are using Gallo. So let's start. We know that we are facing intensive change in the international demands in the market for the livestock system in general. We know that we need to increase productivity as population is constantly growing, and we expect to have to increase increasing consumption up to 50% by 2050. But we also have a high demand for quality, welfare, and sustainability in this production chain. And how we can achieve all these goals at the same time, knowing that the supply growth will be mainly based on the improvements in farm management and animal productivity and less number of lactating cows, as we are facing also reduction in the available lands for livestock. One of the key points in this process is the improvement in feed efficiency. We know that managing the decisions that are related with feeding strategies can account for a large proportion of all variable costs in Earth. Some literature, some reference say around 75%. This can change. But what is the definition of feed efficiency? Feed efficiency, or a more efficient animal, can be defined as an animal that is going to eat less and produce the same or a higher amount of final product. This final product can be milk, can be meat, can be any product that we can obtain from this animal. And we also know that animals that are in this range of a higher feed efficiency, they also have some genetic correlations with higher welfare values and also a reduction in emissions and other important traits for the environment. We have several metrics for feed efficiency in general, but two of the most frequent that we can see in the literature are the feed conversion ratio and the residual feed intake. The FCR, the feed conversion ratio, can be defined as the relationship between dry matter intake and the energy-corrected milk. The dry matter intake is how much the animal eat in a very simple way, and the energy-corrected milk can be estimated using different formulas, but pretty much is a relation between the milk yield and some components in the milk, such as fat and protein, okay? And also the residual feed intake is another very common metric for feed efficiency. And residual feed intake is a little bit different from FCR, as residual feed intake, as the name shows per se, is the residual of a regression, a residual that is obtained of a regression that estimates the intake of the animal. So we can use different formulas to compute the dry matter intake, also using in most of these formulas the energy corrected milk and some change in the weight during the period that we're estimating, and then you can obtain this metric, so the error of our estimation. Okay. Regarding the genetic basis of feed efficiency, we know that in sheep, some meta-analysis and also estimations of irritability show that for RFI, we have irritability that goes from 0.10 to 0.45, and for FCR from 0.05 to 0.77. So a higher irritability for FCR. And knowing this, we know that it's possible to select animals to produce a more efficient progeny. However, we also have some difficulties related with this process. To estimate the feed efficiency of an animal, we need to breed this animal, test this animal for a specific period, and then estimate the feed efficiency. And this is associated with cost and work, and also delay the process to select the animal for breeding. And on top of that, we have some specific issues with each one of our metrics of feed efficiency. For example, FCR. FCR is shown in the literature that has a negative correlation with growth. More feed efficient animals tends to be bigger, and this is an undesired hitchhiking effect. Also, FCR per se is a ratio, so we have an unpredictable selection if we don't know how these components are proportionate or not. Okay, and also is a nonlinear measure, which makes it a little bit more difficult to fit in the regular models. Residual feed intake, as I mentioned, is the error of a prediction, so relies in the performance of a model, and a model that usually is composed by multiple fixed effects, so can suffer for overfitting, sometimes due to the difficulty to obtain all the observations of an animal for the dry matter intake or the other traits used in the estimations. We don't have a very large sample size, and this can affect the precision of this model. In this context, the identification of biomarkers with a good predictive potential for feed efficiency could be useful to reduce the costs and improve the selection of animals with higher feed efficiency. As with these biomarkers, we could easily access these animals in the population and apply a faster selection. Okay? However, this process is not simple. We need to define at which level of information we're going to search for biomarkers. Feed efficiency is a very complex phenotype that is a result of the interaction of multiple biological levels and also the interaction with the environment, and sometimes it's very tricky to try to identify biomarkers. Two of the levels of information that we are doing research here in León is on the epigenetic level and in the metabolic level. So we are investigating the epigenome and also the metabolome of some animals and also other kind of omics, but today I'm going to focus on these two. These two omics data are interesting by some specific characteristics. We know that the nutritional status can directly affect epigenetic alterations in the animal and also vice versa. And nutritional effects can affect actually enzymes that are related with the regulation of the epigenetic process, what also is a response of the first statement. And we know that the epigenetic markers can be irritable, but can also undergo of adaptations through environmental changes or challenges or situations that result in the effect over these enzymes related with the epigenetic process. On the other side, we have the metabolome that is the individual nutritional status of an animal can be explained by a metabolomic signature. And a very interesting characteristic of this kind of data is that the metabolite is one of the final products of a very complex regulatory network. So we are kind of measuring the final effect, the closer final effect in the omic level. So it's also is relatively easier to measure compare, for example, with epigenetic alterations, and can give us specific details about the physiological condition on a specific period of the animal. And in this context, we design an experiment trying to evaluate the effect of feed restriction in some animals. We try to estimate the effect of a reduction in protein, and for this we use 40 ASAFIs in the pre-pubertal age, before 3 months, where we reduce the protein by 16% of the total protein, and we divide these animals into groups of 20 animals. By 64 days, they were fed with a controlled diet, a regular diet, and a diet without this crude protein. So after this period, the animals were get pregnant, and also we took samples from the limbs of these animals at the end. We have some additional studies that are already published using the same experimental design, show the international impact of this diet protein restriction in the limbs, also evaluating the transcription, the expression of long non-coding RNAs and general expression in the perirenal fat of the lambs. But today we're going to focus on the eels and the feed efficiency of the eels. So these 40 eels were subjected to a challenge, so a period of monitoring for all the dry matter intake by 3 weeks, where they were receiving feeding ad libitum, and we were measuring daily records for dry matter intake, milk yield, and weight. Okay, all this data is available on a GitHub page, where you can see all the estimations of the FSCR and RFI, and also the formulas used for these estimations. What is a very interesting data set also for learning and teaching. Okay, so we estimated RFI and FCR using two standard formulas for ASAF. I'm not gonna enter in details for this, but if someone is interested, I can explain later. And we measure the feed efficiency in the first and the second lactation. Okay, so in the first lactation, we're able to get data from 39 eels. In the second lactation, 21 eels. We lost some eels during the first and the second lactation for reasons such as cooling or health problems. Okay, we took samples in these two lactations for to perform untargeted metabolomics and whole genome bisulfite sequencing. Here I'm present the results of the first lactation for the metabolomics and the second lactation for whole genome bisulfite sequencing for the 39s and 21 use respectively. Focusing on the metabolomics, as we had a larger sample size, what we did, we divided the sample in 3 groups: high, medium, and low feed efficiency. This for the both metrics, RFI and FCR. Okay, and we ended with this 10 animals as high feed efficiency, low as 10 as low feed efficiency, and 19 in between. We can see that the means between the groups are different. So we have kind of a good separation of animals in each of the groups. And using the 3,000, almost 4,000 features that I'm calling here, but also can interpret as a metabolite. We cannot call necessarily a metabolite because we need to annotate this first, but we have 39, almost 4,000 features from the metabolomic design. I also have some slides here if someone is interested with the pipeline of annotation and also the high-gel LPOC for detecting the metabolites, but let's focus on the methodology. With these 4,000 features, we apply a partial least square discriminant analysis where we were able to separate the groups of animals that we have, the high, low, and medium RFI, with a good precision. We know that we have some animals that are kind of the edge between the separations, but the Hawk curve was quite interesting for the separation of these three groups. And using the Mixomics package on R, we were able to identify which metabolites were explaining better the separation of these groups. So they were accounting for the higher percentage of this variability. And for FCR, we were able to identify 26 metabolites and 41 using RFI as the feature for this discriminant analysis. When we check the enrichment analysis for these metabolites for this 41 and for this, 41 and 26, we can see that in general, the enrichment of the biological process and actually here the metabolic pathways for these metabolites are associated with the metabolism of sugar, metabolism of carbohydrates, gluconeogenesis, lactose, fructose, what makes a lot of sense when you think about the sample that we're analyzing, that is milk sample. So this is very nice to see when we check the enrichment after selected some specific metabolites. And what we did with this 39 ASAF EOS, we returned to the feature quantification step. We got the values of quantification for these 39 metabolites, and we build 4 different scenarios. A first scenario where we use all the 3.7 thousand features to predict RFI, the same to predict FCR, the 41 selected features based on the VIP values in the Mixomics to predict RFI, and the 26 FCR. There is a mistake here. And we use 5 machine learning algorithms, neural network, random forest, support vector machine, extreme gradient boosting, and orthogonal partial least square to predict the feed efficiency metrics in each one of these 4 scenarios. For the whole genome by feed sequencing, as I mentioned, we only have the 21 EOS in the second lactation. We apply a very standard DML and DMR pipeline detection. So we start with BS seeker to align and to perform the methylation calling, and using the DSS package on R, we identify the DMLs and consequently the DMRs. The DMRs are just a concatenation of sequential DMLs that we have in some specific regions of the genome. And we did this applying some comparison of groups, so high and low RFI in this case, 7 against 7, 7 against 7 for FCR, but also as we had here a lower sample size, we decided to create a new group that is a consensus group. And what this means? This means that we have animals that were classified as high and low RFI for both metrics, and we end up with a 6x6 design here. After this step, we create the model to identify the DMLs and the DMRs. As I mentioned, just the consecutive DMLs are merged in the DMRs. And we got the mean methylation level within each DMR. And we build other 4 scenarios. A scenario where we use the means of methylation within the DMRs identified for RFI to predict RFI, the same for FCR, and to predict the RFI and FCR groups, RFI and FCR metrics in the consensus groups, we use the mean methylation in the DMRs identified for RFI and FCR. So we have more 4 prediction groups, and we use, in this case, 3 machine learning algorithms. The same neural network that I mentioned before, random forest and extreme gradient boosting. Why only 3? Because in this point, we have a much larger sample size for the features, and it takes much more time. So we decided to focus on some specific algorithms here. Okay, so just showing here the separation between high and low. You also have a very good separation when you check the means, and you also can show that there's not effect of the nutritional challenge over the separation of RFI and FCR in this case. But we also were interested to evaluate genetic variants within the differential methylated regions, the DMRs. And what we did, we apply a new pipeline that's applied using a software called Revilio, where we can use the whole genome bisulfite sequence data to perform a pipeline of variant calling. So we identify all the genetic variants within the DMRs that we selected in our comparison groups, which at the end we end up with other 4 predicted scenarios that are the same ones that I described before, but this time including the genetic variants, the SNPs, mapped within the DMRs. So just to contextualize about functions of these DMRs, when we check the most significant DMRs for RFI, FCR, and consensus, we can see that the enrichment for biological process are associated with some metabolic pathways, such as the apelin signaling pathway that is associated with the feed of the animal, growth hormone synthesis of cytosine, lipid metabolism in general, which also makes sense when you're talking about feed efficiency. And how we did the predictions, as we are in a very limited scenario talking about sample size. We decide to evaluate the overall performance of our models, applying some randomization of our training and test sets. So we apply 100 random interactions where we split our data in 17% training, 30% testing. We apply the hyperparameter tuning in the 17% of the data set, applying the best model the best hyperparameters in a model, and we evaluated the selection of all 100 models generated based on the maximum Pearson correlation with the original feed efficient metrics. So at the end, for each one of the scenarios that we create, so here 12 for metabolites, 8 for DMRs, we selected the best model using a ratio between the Pearson correlation and the root mean square error. So we could check the performance of this best model. And we also estimated the mean of the 100 model generated applying this random assignment of animals in training and test sets. Result of metabolites, we could see that for metabolites and RFI and FCR, actually, the results in the bottom of this slide in the left-hand side, using the 4,000 features or the subset of features that that were explaining better the variation in our classification, we didn't improve too much. We have a very nice mean for the prediction, Pearson correlation of 0.78. But for RFI, this scenario is not the same. For RFI, when we use all the features, we have a much worse performance when we use the subset, which could suggest overfitting of our model for RFI. And for FCR, the best model in the two scenarios was the orthogonal partial least square, and for RFI, using all with a very bad performance was XGBoost, and the neural network was the best model when we use the subset of metabolites. These results are kind of similar when we check the root mean square error. The error is kind of lower for the metabolites, but when you compare the absolute values and the magnitude of the values, they are very similar, you can see, but we cannot compare directly the values, as the root mean square error depends of the scale of our values, and the scale of FCR is totally different from the scale of RFI. So even with 14 being equal 14, root mean square error of 14 for FCR doesn't mean the same as a error of 0.14 for RFI. The results of this study are published in a paper in Journal of Dairy Science, and as a general conclusions, we know that FCR show a better prediction performance than for RFI. This is consistent with what we see in the literature in general. We have a slight difference between machine learning algorithms, not anything very significant, with the orthogonal partial least square being seen to be the best option for FCR and the neural network for RFI, okay? And when you talk about whole genome bisulfite sequencing, we have a very different scenario. In general, the predictive performance is much lower. When you talk about the means of the 100 models that we generate, the best mean that you can obtain is 0.08 for FCR, this including the genetic variance that we have there. Sometimes we have even a negative correlation, okay? And here, the thing is that when we also compare the errors, we saw interesting difference for the errors when we use the comparison for the consensus and when we use FCR and RFI individually. Usually for consensus, we have a higher error. So it seems that it's better we use each one of the metrics individually to separate the animals, okay? In general, we have a much worse predictive performance for whole genome bisulfite sequence in the prediction of FSCR and RFI values. Again, very slight difference between machine learning models, and the inclusion of the genetic variants in our predictions seems to improve our accuracy, what actually at the end is not good at all when you see the means. But even in the worst scenarios, when you check the 100 model generated for each one of our predictive scenarios, we can find models that performs very well. So models with correlation of 0.86, 0.73. But when we compare with different compositions of animals in the training and test set, we have a very bad overall performance. This can suggest us that with a better sample size, with a more precise definition of FCR and RFI, maybe epigenetic data can also be used for prediction. But we need a much better sample to try this, okay? Moving forward, I'm going to start to talk a little bit about production now, but I need to do a contest about the production scenario of the Spanish sheep dairy industry. Spain is the fourth milk producer worldwide. Usually in the genetic selection, milk yield and solids are the main goals, but usually the main intent is the cheese production. And among all the breeds that we have in Spain, we have a SAF that is one of the main dairy breeds, and we are studying the effects of different different challenges over the production in ASAF. And here today I'm going to focus about a study of pleiotropic effect for cheese production traits and dairy traits in the ASAF population, and also the inclusion of somatic cell scores, the health component of this pleiotropic effect. I mentioned this before, we need to reach all these levels of improvement simultaneously. It's something difficult to do. And we also know that production and adaptation are intrinsically related in livestock species. Usually, the livestock species are selected for a higher robustness that after this can be selected for higher productivity, and animals that usually are more robust tend to produce more in some challenge scenarios. And we know that understand this relationship between adaptation and productivity is something crucial in our current scenario of climate change and also with constant environmental change for the animals. And we reach a very interesting level of specialization through years and years of genetic selections for several populations, where we have very specialized animals that can produce a lot in very specific situations. So with this change in the worldwide and environmental scenario, we also need to pay attention on animals that can be less susceptible to this change. So animals that will be more consistent in the production and suffer less with these change. Animals that we call resilient. Okay, I'm going to talk a little bit more about resilient in the next slides, but just to start this contextualization. And in this scenario, the evaluation of health and welfare traits are important to select animals that will be more resilient. So we need to also pay attention in different and new phenotypes to be included in the selection of these animals. But selected for multiple traits at the same time is a very difficult task, and we need to know and you need to understand the genetic correlation among these traits to avoid undesirable hitchhiking effects, as I mentioned some slides ago. And in ASAF, we have a moderate to high correlation when you talk about milk traits, cheese production, and somatic cell scores. Some strong negative correlations, for example, between the log of somatic cell scores and protein, fat, and dry extract percentage in these animals. So we were trying to understand a little bit more the pleiotropic effect observed between milk production, cheese production traits, and somatic cell scores in the ASAF population. But what is pleiotropic effect? The pleiotropic effect is when we observe the genetic locus contributing for the expression of two or more phenotypes simultaneously. Okay, so we have this in this scenario, the associations between, for example, the correlations between milk production traits, log of somatic cell scores, and also the cheesability traits. And an integrative approach where we combine multi-omics investigation in a system biology perspective can be a very interesting approach to try to identify the genetic basis of these genetic correlations and understand the biology of the pleiotropic effect observed across these traits. However, we know that integrate omics data is not so easy. Actually, it's a very difficult process. We need to deal with different challenges. Usually, we are working with multidimensionality problems and nonlinear relationships when we start to pile the data. So usually the relationship between the metabolome and the transcriptome and the genome will not be linear. We have curse of dimensionality problems. Usually we have much more variables than observation points, much more data generated than animals analyzed. We need specific tools to deal with these problems. And An interesting alternative to deal with this problem is the application of machine learning as well, but also representative learning approach. And here I'm showing an adaptation of a pipeline proposed by Wu et al. In 2018, where a hierarchical model is applied to combine gene-level p-values and network embeddings to prioritize candidate genes. So with this approach, we're trying to identify the most interesting functional candidate genes for the pleiotropic effect observed between milk production, cheesability traits, and somatic cell scores in dairy sheep. I'm going to talk about briefly about each one of the components of this model. But first, we need to identify regions in the genome that have a potential pleiotropic effect for all these traits. And here we are using a population of 1,039 ASAF eels that were imputed to high density using a reference population of ASAF artificial insemination rams. So the 47 most important rams for this population. So we got a very interesting accuracy in the imputation, even with a limited sample size of the population of reference population, because they were genetically correlated, closely genetically correlated. We apply genome-wide association studies for 12 different traits, 5 milk-related traits, 6 cheesability traits, and log of somatic cell scores. And just briefly speaking about these traits, milk yield, fat percentage, protein percentage, the fat and milk yield. And we have some additional cheesability traits such as the time for coatling, the log of the period, the log of the coatling estimation for these animals, and also the yield of the cheese produced at the end. So using the GCTA software, we apply a linear model where we use daisy milk, age at parturition, flock test day, and number of the lambs as variables in this model to perform these GWAS. So we end up with a very large matrix of all the variants that we include in our GWAS and the 12 traits that we analyzed. And using the multi-trait test statistics proposed by Bolomeyer et al. 2014, we estimate the potential pleiotropic effect for each one of these markers. Briefly speaking, in this strategy, we use this N by M matrix here in the rows N, all the markers that we have, M, the T statistics for the association test that we have, and using a chi-square distribution after conversion of this matrix, we can estimate a potential p-value of this pleiotropic effect. Okay, so we're going to have at the end a vector with the length equal the number of markers. But as I mentioned before, in this hierarchical model, we need gene level p-values. And how we can estimate p-value in a gene level if we estimate p-values at the marker level using the previous approach, the GWAS and the multi-trait test statistics. We apply the Davis algorithm, very old and standard algorithm for the concatenation of p-values here. And how we did it? In this pipeline, we use Gallo to annotate all the markers from our GWAS and that were subsequently estimated the pleiotropic effects within our genes using our 100 kb interval. So all the genes in the genome we compare with our GWAS. And we also, for each gene, we estimate a matrix, a linkage disequilibrium matrix for all the markers mapped within this gene. And in the Davis algorithm, we estimate the p-value at the gene level as the sum of the products between the lambda, which is the eigenvalue for this square linkage disequilibrium matrix. So we decompose this linkage disequilibrium matrix within the gene. We obtain eigenvalue for this linkage disequilibrium matrix, and we multiply by a random variable that is based on a non-central chi-square distribution. And at the end, we obtain a p-value for each gene individually. Sorry. And we also need the functional part of our model. And the functional part of our model was based in some network strategies, okay? The first network strategy was based on the transcriptome of this animal, so using 28 ASA fields that we have RNA sequencing data. We use WGCNA to identify co-expressed networks in the milk transcriptome. We were able to identify 10 co-expressed models of genes, and for each one of these models of co-expressed genes, we will obtain the topology matrix, so a matrix that shows the associations between each component of this network. Using these 10 co-expressed models, we apply GO and pathway enrichment analysis individually for each model using the GIF Profile 2 package. And we did this for gene ontology for biological process, molecular function, and cellular component. And for pathways, we use KEGG and reactome pathway. We apply redundance reduction, and using a network representation, we feed our model with this data. So we have a matrix that shows the association of each gene with each term, if the term was enriched or not. And we also apply KTL annotation with each one of the genes using Gallo and a 100 kb interval. And we use the same representation. So if a gene was mapped in a region with a previous described KTL, we have association between this gene and the KTL in the matrix. But we have now several matrices that are very large in dimensions, and we cannot fit this directly in our model. So we need to reduce this dimensionality. And how we did this? We did this using the Node2Vec library on Python, where we applied a network embedding approach using a random walk, and we could reduce the dimensionality of our data, that is a network, to a matrix of 32 dimensions. So each gene is represented as a row, and we have 32 columns that represents the connections in the networks, okay? But again, feed a network with 32 dimensions is also difficult in a model, so we need to reduce even more. And using the embeddings that we got from the WGCNA, the gene ontology, the pathways, and the KTLs, we use a linear discriminant analysis that reduced these 32 dimensions for a single dimension vector. So we got a vector that multiplies at the end our 32 dimension matrix, and we can obtain just a single vector that represents the in a very short way the importance of each gene within each one of the networks that we have. And applying a hierarchical model with the combination of the gene level p-values and the importance of the genes within each one of our networks, we can fit a hierarchical model that through expectation maximization approach can estimate the posterior probability of association of the gene with our hypothetical phenotype, that in this case is the pleiotropic effect. Closer from 1, higher the association, the probability of association with the pleiotropic effect. We obtained 15 candidate genes that had a higher, that has the higher posterior probability of association in our data, PPA higher than 0.99, and gene level p-value is smaller than 0.05. And among these genes, we have some very traditional genes for milk production and cheese, such as the kappa-casein, the vitamin D receptor, some solute carrier receptors, that is the ACL the ACSL1 gene directly associated with the control of the lipid metabolism that kind of functionally validate our approach. But the most interesting thing is that when you got these 15 genes and we perform pathway enrichment analysis, we can cluster the pathways that are enriched for these 15 genes in three main groups of pathways. Pathways associated with the mammary gland development and lactation, membrane transport, mainly for fat acids and carbohydrates, and lipid metabolism. This is really interesting because they are directly related with milk production, but also production of specific components in the milk, and also the transport across membrane that is also important for the cheesability traits, and also in the defense against mastitis, for example. And among these genes, as I mentioned, we have vitamin D receptor that is directly related with the immune response, but we also have the PTGR2 that is not directly related in the literature with the pleiotropic effect, but we know that the PTGR2 is a important regulator of the PPR gamma gene that acts activating this gene, and we know that PPRG gamma is a key component of the pleiotropic effect observed between lactogenesis, control of inflammation, and adipogenesis in the mammary gland. So with this approach, we could help in the unraveling process of the genetic causes between the genetic correlations observed between milk traits, cheesability traits, and somatic cell scores in the ASAF population. We found these 15 potential candidate genes, and we know that they are acting in the mammary gland in specific biological processes that are important for the traits that we're evaluating. However, we need to explore other scenarios, prospect for potential genetic markers, and perform cross-validation. Currently, we are trying to apply the same pipeline in a much larger population with other traits for the ASAF here as well. And just briefly, I will talk about resilience. I think that I don't have a lot of time, so I will not, maybe I will not talk about the Gallup updates today, but it's not a problem as I mentioned. One way to try to cross-validate these regions is try to look for the association between production and adaptation for another perspective. As I mentioned, resilient animals are really interesting for the current scenario that we have in the production chain in the livestock sector. Resilience in the dictionary can be defined as the capacity of withstand or recover quickly from difficulties. And animals that usually are sensitive, they will show less disturbance in the expected observations in the production records for this animal. And usually animals that are higher, that has higher genetic merit for productivity tends to be more sensible for alterations in the environment. This is we can see here, for example, when we compare in cattle, the genetic traits for milk yield and the slope decay for heat tolerance across the years. And you can see that a clear pattern of opposite pattern here. And the resilient animals might be animals that are in this intersection between the specialized animals and the generalist animals. We still want animals that produce a lot, but we also want animals that has a higher potential of resilience, a higher potential of adaptation. So if we try to understand the associations between adaptation and productivity and how this can be interchangeable, it's a very interesting approach. And a classical approach to find regions in the genome for any kind of trait is the analysis of selection sweeps, regions in the genome that show difference in the allelic frequency after a selection period. And maybe if you try to find regions that are commonly observed when we do studies for selection sweeps productivity and also adaptations, we can try to understand better this relationship and also find pleiotropic regions that might be used to select more resilient animals. The results of this study is published in a paper that we recently published in the Genetic Selection Evolution. And what we did a systematic review for selection sweep studies for adaptation and productivity in sheep. So we end up with some traits for adaptation, some traits for productivity, thermoregulation, resistance to parasite, fat deposition, and high altitude. I put fat deposition as adaptation because we're mainly talk about deposition of fat in the tail that is associated with resistance of some extremely hard environments with lack of water and high temperatures, and milk yield, wool, meat, prophylaxis, and litter size. And we end up with 23 studies for adaptation and 15 for production. Okay, applying the study of selection sweeps. So the 38 studies, we got all the coordinates of all markers. We translate for the Rambouillet version 2.0 reference genome, and we annotated all the genes that are in. Okay, sorry. So if I have a coordinate instead of a SNP, so if they show us a window, we convert exactly the position of this window. But if we use a SNP, we use the approach using a 250 kb window, okay? In the moment to annotate the genes. So what we did, we checked the overlapping among the studies, and regions where we have at least 3 selection sweeps overlapping in the genome, we call a confirmed selective sweep. And using this confirmed selected sweep, we classify them in production or adaptation selection sweeps. Production, those confirmed selection sweeps where more than 60% of the SS mapped there were coming from production studies, and adaptation, those with 70% of the selection sweeps coming from adaptation studies. And we use Gallo to annotate genes. And using the annotated genes within the production, adaptation, and the shared selection sweeps, we apply gene ontology and KEGG enrichment analysis using the gProfile2 package on Gallo. And we also use gProfile2 package in R. And also using Gallo, we annotated the KTLs and applied KTL enrichment analysis for these 529 confirmed selection sweeps, 213 for production, 122 for adaptation. We create an incidence matrix between the genes and the gene ontology terms, and we identify the most important genes in this network using betweenness centrality. And then we apply again a network analysis to try to understand the relationship between the traits, KTLs, and gene ontology terms. Briefly speaking, when you talk about the hub genes for production, CSS, so the confirmed selection sweeps for productivity, we have enrichment results for the development of the reproductive system, feeding behavior, response to temperature stimulus, among other. But here I would like to focus the CNR1 and the GSR genes. CNR1 acts in the cortical neurons, showing a higher increase after fasting, so they can control, for example, the feeding behavior of the animal. And the GSR1 encodes the ghrelin receptor. That is a receptor that is directly related with the hunger of the animal. Also, with production, we have several genes associated with the regulation of the temperature stimulus, okay? And for adaptation, we have response to light stimulus, regulation of growth, and response of growth stimulus. And when we check these genes, the genes associated with these enriched terms, we can see genes that are associated with the control of the circadian clock and seasonability of these animals that are highly important for the adaptation, how much the animal is going to adapt it for the challenge in the environment. And when you talk about regions that are that comes from both studies, production and adaptability, we have a very interesting enrichment for metabotropic receptors for both classes, ionotropic and metabotropic, that are responsible for the modulation of inflammation and pain. And this regulation between inflammation and pain can be interface between disease resistance and productivity. As we know that in the equation of energy dispense in the organism, if animal needs to recover, he needs to take energy that was gonna be directed for productivity and put on recover. So this modulation between pain, between inflammation, can be a key point of animal have a higher productive level and a higher resilience profile as well. So I'm going to be short with the conclusions. We could find some interesting interfaces between adaptation and productivity. The adaptation CCS was mainly associated with seasonality, circadian rhythm, thermoregulation, the production with feed behavior, reproduction, and cellular differentiation, and modulation of inflammation and pain when you check CSS share between both classes of studies. This might help us to improve the fine mapping and fine genomic regions, controlling this interface between productivity and adaptability. And the identification of this region can help us to prospect genetic variants, controlling resilience in sheep population. Okay, I think that I don't have much time, so I will stop now and I'll update it for another time. And if someone is interested, can also ask me or email me that I will be more than glad to answer. Okay, thank you very much.
Perfect. Thank you, Dr. Fonseca, for your presentation. That was very interesting. We have time for maybe one or two questions, so I'll open it up to anybody in the room or online that wants to ask. Oh, Saranya?
Hi, Pablo.
Hi, Saranya. Nice to see
Hi,
you.
how are you? After a long time.
Good.
I'm just wondering because I see that you have used this machine learning to predict the RFI and the FCR thing. And then I see that you are using like nearly 100 models, and then you are choosing the one with the high correlation and with the low RMC. But I'm just wondering, like, even when you are predicting with like 100 models, did you look at the frequency of those correlations, the models being predicted again and again? What we find is like the model that has the highest correlations may be predicted maybe 1 or 2 times, but some of the models are predicted high frequently. Have you looked into that?
This is what we checked. This is what we see when we check the mean of our of our accuracy metrics in the 100 models. We can see scenarios where the prediction are quite good, but with a specific subset of animals in the training and in the test population. And this can be an effect of directly of our sample size that's very reduced because we have 38 animals. And the idea of these studies is just to have a start point. We want to see if you have a potential of prediction, maybe select some variables that you can expand in a larger sample size to really estimate the accuracy. Okay, and we know these kind of models, they can be affected by overfitting, they can be affected by the composition of our training and test population, and you need to be careful with the interpretations. Okay? But anyway, the results show some interesting applications and potential applications for these methodologies. I don't know if I went directly to your question, but if not, you can tell me.
Thank you.
You're welcome.
Thank you. Flavio, did you want to ask a quick question?
Yes, and might not be quick. But anyway, Pablo, good to see you. Thanks
Good to see you, Flavio.
for the presentation. Very interesting, lot of information. So we should have more time for discussing. But a very quick question for understand the first part of the presentation about the methylation. So which tissue did you use?
Milk somatic cells.
So I think you never mentioned, so I was not sure. So milk somatic cell, okay.
Yes.
And then when you you had your variants, the methylation, right, markers, and then you included the variants from the genome.
Yes.
So how did you look at the collinearity that you are introducing when you did that?
Yeah, the collinearity there can be an important issue. But we test the models without the variants, and we were getting very low accuracy and very low performance, and we decided to include the variants to see what might happen there. So we didn't get any relevant increasing of accuracy there, but definitely we have a high collinearity there. This might be a problem for example, generalization of this model later. But one thing is that we are also trying models fitting only the genetic variants. The idea to include the genetic variants is because it's much easier to estimate to apply a model with a genetic variant than with the mean methylation level within a DMR. So even if you have a high collinearity and you can have similar results breaking the predictions for only the DMR means, and only for the genetic variants, probably we should shoot for the genetic variants within the DMRs. Easier, cheaper, and faster estimation of these variants to feed the models.
Yes. I would expect some level of collinearity, right? So that's. And my last question, just to understand, Paul, you showed in a slide that you use as a criterion the square of the correlation, the ratio of the square of the correlation, the Pierce correlation, over the root mean square error.
Yeah.
What is the interpretation of this ratio?
Okay.
I don't understand it very well.
Just one thing is that it's not a square, just not a square. This was a typo. It's just the Pearson correlation. It's just a Pearson correlation there.
Yeah.
It's because I tried to do there is because for some specific reason I was also for a specific reason, no, for the reduced sample size. I was having some scenarios where I had a high Pearson correlation, but with a different error. For example, I have two scenarios with the same Pearson correlation, but with very different errors. With very different errors in the magnitude of the errors. So I decided to do a ratio between both to try to compensate these differences. So this was the first approach that we tried. But
I think probably if you had that, Pablo, probably because it's because of the bias, right? Because the mean square error is capturing, in addition to the prediction error varies, the bias, right? So I think in that case, the model has some level of bias. That's why now you're seeing very different, much bigger mean square error than the correlation. It's very well correlated, but there is a bias. It's not on the same scale, the prediction.
And this is something that I didn't mention in the presentation, but we have in the paper, this bias we see more frequently when you feed the model with the complete data set of features. When you have the pre-selected subset of variables, for example, for the metabolites, this bias is substantially reduced. So probably this bias is caused by some combination of a reduced sample size and overfitting of the model.
Yeah. Okay, so that's a great presentation. I would have much more, many other questions, but I will stop here, see if it's somebody else that likes to ask a final question. Thank you so much, Pablo.
You're welcome, Flavio.
Thank you. Unfortunately, we're out of time today, but I did want to thank you, Dr. Fonseca, for presenting to us today. That was a really great discussion. The recording will be posted to YouTube later, and we'll send you those details. And thanks to everybody for joining. We'll see you next week.
Thank you very much.
Thank you, Pablo.
Thank you, Pablo.
Thank you, Angela.